Okay, so, well, Kjirsten Wayman, who is an illustrious organic chemistry professor, and so therefore must be very, very smart in many ways, will be telling us about the scents, that is the smells, of various species or forms of plants in the group called the Western sessile trilliums. And she just taught 6 hours, and she's going to project her voice so that the back can hear. So please help me welcome Kjirsten to the podium.
All right, thank you. So as the title suggests, I'm going to talk about the floral scents of Western sessile trillium. And what motivated this project is the fact that the Western sessile trillium can be very difficult to distinguish, very difficult to identify. And one of the reasons is that they have very few traits. And within each of the species, there's lots of variability of those traits. And then there's also lots of overlap amongst the species for those very same traits. So there's very little to go off of in order to distinguish these species and identify them. So what I was hoping is that we could look at the floral scent compositions and see how the floral scent compositions for the various species differ. And if so, can we use the floral scent compositions to help us distinguish among the species? So before we talk about the species themselves, I need to talk about this word sessile here. So many of you are probably familiar with Trillium ovatum. Can you see the cursor? Yeah, good. Okay, so many of you are probably familiar with Trillium ovatum, this species here. It's in the group of Trillium that we call pedicellate Trillium. So we broadly classify Trillium as either pedicellate or sessile. And the pedicellate Trillium have flowers that are borne on a stalk. So this flower is attached to the rest of the plant by a stalk here that we call a pedicel. And in this side view here, you can see that stalk a little bit better. Whereas in the sessile Trillium over here, notice that the flower is not attached to the rest of the plant by a stalk. There's no stalk there. The petals, the sepals, the ovary, the stamen, all those floral parts are attached to the same part of the plant as the leaves are. So we call this a sessile trillium. And down below here, I have another species of sessile trillium where once again, you can see that it lacks a stalk. So these are the sessile trillium, sessile-flowered trillium. So I have listed here all of the sessile trillium and pedicellate trillium in Western North America. And once again, we're going to focus on the sessile trillium in this talk. So there are 7 taxa, including subspecies and varieties of sessile trillium in Western North America. And of those, 5 of those taxa occur in California. And of those 7, we're only going to be focused on 6 of them. So the last one listed there, Trillium petiolatum, is not part of this study, and I'll explain why in a short bit. So the rest, Trillium albutum and its subspecies Parviflorum, Trillium angustipetalum, Trillium chloropedalum and its 2 varieties, Chloropedalum and Giganteum, and then also Trillium courbiaceae. Those are going to be the focus of the study here. So the most recent revision of the Western sessile trillium was done in 1975. That was quite a while ago. And this was done by Freeman. And in this map here, this is the map that he published with his revision. And so I'm going to go through and just mention the regions of the species. So I've outlined here up in this circle are the plants of Trillium petiolatum, which is the one that's not part of this study. Trillium petiolatum occurs in Idaho, Washington, and Oregon. Trillium albutum here in the large blue circle occupies the largest geographic range, and it extends all the way up into Washington, down into towards the Bay Area, and then over to the Sierra Nevada foothills. So it has quite a range. Now, up here in Washington, after Freeman published his revision, more in the, I think it was 1980s when Victor Soukup published the fact that he thought that these plants up here were different species called Trillium parviflorum. There's still some debate as to whether or not they're separate species or whether they're just a subspecies. And I officially call them subspecies because that's where the taxonomic treatments lie, but I might just call it Trillium parviflorum today as well. But they exist up here in Washington. How far down into Oregon they go is still unclear, but it's still all part of the Trillium albidum group. You might know Trillium Kurobayashii, so that occurs here in northwestern California and also in southwestern Oregon, so this small area here. Freeman also indicated that these plants over here in the Sierra Nevada might also be Trillium Kurobayashii, although my feeling is that they're just an extension of Trillium angustipetalum. So Trillium angustipetalum occurs here in the Sierra Nevada foothill and also down here along the coast of San Luis Obispo County. And then right here around the San Francisco Bay areas where we see Trillium chloropetalum. And there are two varieties of Trillium chloropetalum. One is variety chloropetalum and one is giganteum. And so they're all nestled in there together. And then a couple populations here in Napa County. To me, they look like angustipetalum. Freeman indicated they might just be chloropetalum, and we'll discuss those as we go through the talk a little bit more. So I'm going to first introduce you to the Western sessile taxa, and we're going to start with Trillium kurobayashii. So Trillium kurobayashii is a plant that has maroon petals. And what you'll also notice is that if we look closer in at the ovary and stamens, we see it also has maroon stamens and a maroon ovary. And I might interchange the words maroon and purple. In fact, I might even say maroon and or purple. So those words are kind of interchangeable today. So once again, we have the maroon petals, we have maroon stamens, and more specifically, it's the anther connectives in those stamens. And also what you should notice is that the height of the stamens is generally a little bit above the height of the ovary to about 1.5 times the height at its maximum. So that ratio is going to be important for identification. And one other thing with Trillium kurobayashii that I don't typically notice with the other sessile Trillium is that sometimes as the flower ages, you can see this twist in the petals. And that seems, at least to me, to be a unique feature of this taxon. So another feature of sessile Trillium is the leaf modeling. So you can see here Trillium kurobayashii with very little leaf modeling in this example. And then the other three photos, we have quite extensive leaf modeling. And from what I've seen, Trillium kurobayashii seems to have the most elaborate leaf modeling of all of them. Now, Trillium albidum is the species with white petals. And in fact, it's the only white-petaled species well, of all. So in Eastern North America, they have a lot of sessile Trillium, but they do not have white-petaled sessile Trillium. So this is unique to our region. So I'm going to describe four different variants of Trillium albidum. And the first one is the variant without purple pigment. So these plants tend not to have purple pigment on their floral parts, although you might find the odd one here and there that might. But typically we see that the petals are all white. If we look at the ovary and stamens, we see that we don't have that dark maroon or dark purple color. In fact, we've got more of a pale yellow to greenish color there. And so both the anther connectives and the ovary are a light color. One thing you notice in terms of the orientation of the stamens don't touch the ovary. They're kind of splayed out from the ovary. And that's to be distinguished from Trillium kurobayashii, where the stamens are pressed against the ovary. So that's one difference there. Also, the height of the stamens is about twice that of the ovary. So that's another distinguishing feature there. Trillium albidum can exist as single plants, or you might see doubles, you might see clusters, and you might even see fields. I think this is the species, the only one where I've, at least in the Western US, where I've seen sort of large areas or fields. The others tend not to do that. So Trillium albutum with purple pigment has all the same characteristics as the one without purple pigment, except for you're going to find purple pigment at the base of the petals. You'll also see it at the base of the stamens here on the filaments. And then also the ovary tends to have some purple pigmentation, whether it's at the tip of the ovary or elsewhere. And sometimes we'll even see some pigmentation in the anther connectives here as well. Other than that, the plants with purple pigment have largely some of the same characteristics as those without. Now, Trillium albutum in the Marble Mountain Wilderness has some unique characteristics. And one thing you might notice is that even though we say this species has white petals, you'll notice just a tinge of yellow in these. And if we compare it to the white petal color of the other two. So there's a little tinge of yellow in these petals. These plants also occur at high elevation. So these are up around 5,500. And even in other parts of the Klamath Mountains, you'll find Trillium albutum up towards about 6,600 feet in elevation. So they can go pretty high. And one other unique characteristic of these plants is that I often find these really large clusters. So I've counted up to about 50 stems in one cluster. And not only that, these plants tend to occur in very sun-exposed spots. So in other areas, you might find Trillium, in the shade of a forest in the understory, or you might see it hiding under a shrub like an alder or a willow. But the plants up here, they tend to be just sitting right out there in the sun in these open meadows. So that's a unique characteristic as well. Trillium albutum subspecies parviflorum, this is the taxon that I mentioned, occurs up in Washington and down to some degree in Oregon. And the unique feature of this taxon is the fact that it has smaller petals than the other Trillium albutum, and they're not just shorter and narrower, but they also have more of a linear shape, whereas the other plants tend to have a more ovate or obovate shape. So the petal shape is different, the petal size is different. And also these plants tend to have that purple pigmentation on the floral parts at the base of the petals, as you can see over here, on the tip of the ovary, and even on the stamens here. Trillium angustipetalum is another maroon petaled plant. And the maroon color of Trillium angustipetalum seems to be a bit darker than Kerbiaceae. It also has the maroon stamens and maroon ovary here. And like I said, that dark petal seems to, or that dark color seems to be a bit darker than what we see for Kerbiaceae. But even down here, these two photos are from the same population along the same trail. These just happen to bloom a little bit earlier. And one thing I notice is that the petal color can often fade with flower age. So that might be responsible for that more red color here as opposed to the dark maroon. Trillium chloropedalum has two varieties. One is variety chloropedalum. And as you can see from this slide, we've got lots of petal colors here. So we've got maroon to sort of this lighter brownish, to bronze, to orange, to yellow, to green. Lots of petal color variation. And in any given population of Trillium chloropedalum, variety chloropedalum, you're going to see every single one of these petal colors. So lots of variation in petal color, and also we'll see in petal shape as well. If we look closer in at the ovary and stamens, you'll see there's a different orientation of those stamens. So even though we have that dark maroon color of the ovary and stamens, notice the shape of these stamens. They're more curved or more bowed. Sometimes they're even curved over the top of that ovary. Sometimes not quite to that extent. They're just bowed about that ovary. They don't touch the ovary like we saw in Angustipetalum and Kerbiaceae. They're curved around that ovary. So very different orientation there. If we look over here at variety giganteum, you'll see a very similar orientation of the stamens. But what you should notice is a difference in petal color. So while variety chloropedalum had more of those greens and yellows and oranges and maroon, giganteum goes from about burgundy to pink to, as we'll see, even white petals. So in a population of Trillium chloropedalum giganteum, we'll see a variety of petal colors from this burgundy color to a dark pink, to a lighter pink, to even white. And I'll mention the white ones in a little bit again. You'll see a variety of petal shapes here. So we've got these taller, narrower petals. We've got some wider petals. Actually, these are giant petals. They're really huge. And then we can even have some more normal sized Trillium petals like what we might more typically see in Trillium albutum as well. In a population of Trillium chloropetalum giganteum, you'll see these petal color variations within a population. So these three photos here, the wider petals, the taller, narrower, and even these pink petaled plants, these are all in the same population, just different parts of the trail. Down here, these two, different petal color and shape, are occurring right next to one another. So we've got lots of variation in these populations. And the same is true for variety chloropetalum. We'll see different petal colors occurring right next to each other. Or we might just see a large cluster of all one petal color. Now, here's a table showing some of the distinguishing traits that we can find for these Trillium. And also, we'll see that a lot of these traits overlap. We're not going to look at the details, so hopefully you can't even see them. I'm going to talk to you in detail about what some of the major issues are in identifying these Trillium. So first, if we're confronted with a Trillium albutum versus a Trillium kerbiaceae or angustipetalum, these are going to be quite easy to distinguish because, of course, Trillium albutum has white petals. It's got stamens that are twice the height of the ovary. They're splayed out and away from the ovary, whereas kerbiaceae and angustipetalum both have maroon petals. Now, one trouble is, how do we distinguish these other two, the plants with maroon petals? Well, Freeman, when he published his revision of sessile Trillium in 1975, he described a variety of these Trilliums. So he described Trillium kerbiaceae and angustipetalum. So that appears in his publication. Flora of North America also took on and recognizes both kerbiaceae and angustipetalum. The Jepson manual, our California plant manual, does not recognize Trillium kerbiaceae as being distinct from angustipetalum. It only recognizes angustipetalum and calls kerbiaceae just a synonym. Flora of Oregon only recognizes kerbiaceae, and they don't recognize angustipetalum as being a species in Oregon. So there's this dilemma as to how many species are here. Is there one? Is there two? And I'm going to explore that answer with floral scent. But before we do that, let's look a little bit more at morphology here. So if we compare directly angustipetalum with kerbiaceae, once again, we see these maroon petal colors. With angustipetalum, they tend to be narrower and more linear shaped, whereas kerbiaceae tends to have wider petals. And again, I mentioned that petal twist that I see in kerbiaceae that I don't see in any of the other sessile Trillium. So that's a bit unique as well. Chloropetalum also has to be thrown into the mix here as well, because Trillium chloropetalum also has these maroon petal varieties. So how do we distinguish it from the other two? Well, one way is that in chloropetalum, we saw that there was that wide variety of petal colors in any one population. So we're not going to just see maroon, we're going to see lots of petal colors. The other way that we might distinguish it is the petal shape. Again, lots of different petal shapes, whereas kerbiaceae tends to be somewhat consistent and angustipetalum also consistent. But we wouldn't have this dilemma of identification if there wasn't also lots of variability amongst those as well. So if we look closer in at the ovary and stamens, we see for angustipetalum, the stamen height to ovary height is about 1 to 1.5. So the stamens can be about equal in height to the ovary, or they can be up to 1.5 times the height of the ovary. The same is true for kerbiaceae. But even though they have very similar characteristics, the stamens are straight, they're pressed against the ovary. You can also see that there's difference in these. So it's hard to put into word the difference when we look at the stamens and ovary of these situations. But even though the height variation is the same and the straightness and how they're pressed against the ovary is the same, there's also some difference in how they appear. But they differ from Trillium chloropetalum. Trillium chloropetalum has stamens that are about twice the height of the ovary. They're curved or bowed about that ovary, and so they are more easily distinguished from the other two. And in addition, we've got that petal color variation in these populations as well. So now comes the Napa County red sessile. So there are a couple populations, one I call Etna Springs and the other is Garnet Creek. These populations are about 4 miles apart by bird. By driving, it's about an hour or so, long distance, but 4 miles by bird. And they have maroon petal colors. They have these long, narrow petals, much like angustipetalum. So we might assume angustipetalum. But if we look closer in at the stamens and ovaries, there's a little bit of variation there. Some are straight and pressed against the ovary. Some have a little bit of curvature, and some have heights that are about twice that of the ovary. So there's some variation there that make it so it's difficult to really say for sure that it's angustipetalum versus maybe chloropetalum. Freeman, in his publication, thought that these might just be monochromatic populations of Trillium chloropetalum. My sense is that they are angustipetalum. So we'll see what Floral Scent has to say about that a little bit later. So another question is with chloropetalum. With all this color variation and petal shape variation, are these really only one species? Are they all just chloropetalum? Or are they two species? Are the two variants two different species? Or perhaps this is one giant hybrid swarm. And if it's a hybrid swarm, who are the parents? Are there two hybrid swarms? Is one a hybrid swarm and the other not? So lots of questions where chloropetalum is concerned, but clearly there seems to be something going on here. And in any given population of chloropetalum variety giganteum, we're going to see a white-petaled variant or two. And that white-petaled variant is going to have stamens and ovaries that are that maroon color, that dark color that's consistent with that species, and distinguishes it from albitum, which has that light color to the ovary and stamens, and also these straight stamens sort of splayed out from the ovary. So there's a difference there. But there are also these plants that have intermediate characters. They've got the white petal color, but if you look at the stamens and ovary, they've got this color that's in between. It's tinged with that maroon rather than being dark maroon or the lighter color. So what's going on in these populations, and what's the identity of these plants? And then with Trillium albitum, I mentioned before there are those 4 variants. Well, are these 4 variants all the same species? Is Trillium albitum subspecies parviflorum a different species? Is it actually Trillium parviflorum? And so the question is, within the concept of Trillium albitum, how many species do we have? And I just want to mention that Trillium parviflorum is the one that occurs in Washington. The extent to which it goes down to Oregon is a question, but it's thought that there's a gradient. So in Northern Oregon, it's thought that they're more parviflorum-like, and towards Southern Oregon, more albitum-like. And so I do have some samples from populations that are in that intermediate zone. And so one question is, which subspecies are they, or are they truly intermediate? And we'll look at some data from those plants as well. Now, I mentioned earlier that Trillium petiolatum was not a part of my study. And one reason, as you can see, is that morphologically, it's very different. There's no way you would confuse this plant with the others. So it's got leaves that are attached to the rest of the plant by these long petioles, hence the name petiolatum. The flower sits beneath the leaves. In fact, the flower rests just about 1 or 2 inches off the ground. And also, these plants smell like rotting flesh. They have a horrible smell. And that horrible smell is to be distinguished from the floral scents of the rest of the sessile trillium that I've discussed. So Trillium albitum has floral scents reminiscent of rose or geranium, lemony citrus, or just a sweet scent. It's hard for me to describe that sweet scent aside from just saying sweet, but sweet is what it is. Subspecies parviflorum has a sweet scent with a hint of clove. That clove is often a distinguishing feature for that subspecies. Trillium angustipetalum has a scent like bananas. Trillium kirbyashii also has a banana-like scent, but it's got a zing to it. I say it's more like bananas soaked in alcohol. And what we're going to find is that's largely due to the fact that there's much greater complexity to its floral scent. And then Trillium chloropetalum can have a variety of floral scents, anywhere from banana to lemon to rose to cinnamon, or sometimes all of that just mixed together. So they've got lots of variation, and we'll sometimes see that variation all in one plant. So the big question is, can we use the chemical composition of the floral scents to distinguish among these species of Western sessile Trillium? So I sought out to answer that question, and in doing so, I sampled from 42 wild populations, and those populations span from Washington down towards San Luis Obispo County in California. At each population, I sampled anywhere between 4 to 27 individuals, and so in the final data set, there are 600 total individuals. And so it goes from, again, from southern Washington down to San Luis Obispo, and then over here even into the Sierra Nevada foothills. So we've got quite a range. So up here, Trillium albidum subspecies parviflorum was sampled. And then we have those intermediates between albidum and parviflorum here in central and northern Oregon. In southern Oregon down to Mendocino County and over to the Sierra Nevada foothills, we've got samples of Trillium albidum. So this spans the widest geographic range of for any of the species. Trillium kerbiaceae was sampled a lot here in northern California and then also southern Oregon. And Trillium angustipetalum, a few populations in the Sierra Nevada foothills, and then down here in San Luis Obispo County. Those questionable Napa County red sessiles here, and then also Trillium chloropedalum, which is all centered right around the Bay Area. And both varieties of chloropedalum and giganteum were sampled. So what is floral scent? Floral scent is a simple to complex blend of volatile organic compounds emitted from the floral parts. And so by simple, it could be, just a few compounds, maybe 3 compounds in that floral scent, or we might have many. In the case of these Trillium, up to 50 compounds can be found. But in other plant species, you might find 100 or more unique volatile organic compounds in those floral scents. Now, for Trillium, at least the Western sessile Trillium, that floral scent is emitted from the petals. So that's the tissue in the floral part that is responsible for the floral scent. In terms of the types of organic compounds, there are a number of classes of organic compounds represented. We've got terpenes, and more specifically oxygenated terpenoids. So citronellol is that compound that has that lemony citrus scent. Geraniol has that rose scent. And we also have esters, so isobutyl acetate we're going to find is responsible for that banana-like scent. We also have sesquiterpenes, apocarotenoids. Those scents are so easily distinguishable, but in terms of the benzenoids, eugenol is the compound responsible for that clove-like scent, and cinnamyl alcohol, you can imagine, has the smell of cinnamon, and benzyl alcohol is a little bit sweet. So these are just some examples of the 92 overall compounds that I determined were in these sessile Trillium. Of those 92, only, I think, 66 were definitively identified. The others still remain unknown or partially known. So how do we determine the number of volatile organic compounds in the floral scent, the relative amounts of each of those compounds, and the identities of those compounds? And the way that I do that is using solid phase microextraction followed by gas chromatography mass spectrometry. And I'm not going to go through the details of these techniques, but I just want to give you a brief idea. So I take a petal from an individual, and I put that petal into a vial or a beaker if that petal is of significantly large size and won't fit here. And then I enclose that petal in the vial with foil. And I allow that petal to emit its volatile organic compounds. And then after about an hour and a half or so, I can insert this needle that has a fiber at the end. And that fiber is made up of an adsorbent material, and that fiber absorbs the volatile organic compounds from that air in the vial. And then after about a half hour or so, I can inject that needle with the fiber into the gas chromatograph mass spectrometer, and that's this instrument here. So the gas chromatograph separates the compounds and tells me their relative quantities. And then the mass spectrometer gives me a mass spectrum of each of the compounds. And you can think of a mass spectrum much like a fingerprint. So mass spectrum is characteristic of a given compound, just like our fingerprints identify us. So I'm going to present the data to you in a couple different forms. And the first form is based on pie charts. So you don't need to worry about what the compounds are, because really they're just going to be represented as colors. So each of the different compounds has a different color. And as you'll see, they're not going to be as crazy as this one pie chart with 92 different pieces. So at any given population, I'll collect a number of petals from a number of individuals. Let's say this example population, I collected 18 petals from 18 different individuals. And then what I do is I analyze the floral scent composition of each of those individuals. So each of these pie charts in this box represents the floral scent composition of an individual from that population. And then I average those floral scent compositions to get one pie chart that it represents the average percent composition of the floral volatiles for that population. So again, each of these compounds is a different color. Now, on this slide, you can see all the different populations represented by a pie chart. And hopefully you can see, as I've sort of grouped them here in terms of their species or various types within each of those species, hopefully you can see that there are some similarities here. So Trillium angustipetalum, we've got some similarities, Kerbiaceae, similarities in colors. And hopefully that you can see that the color patterns that we see in one species are very different from the color patterns that we see in the others. And one thing I want to mention are these two that I've boxed. I just want to mention, I didn't commit to an identification of these. I mentioned that I thought these might be angustipetalum, but they could have also been chloropetalum. What's your guess? They look a lot like angustipetalum, don't they? Yeah, and then if we look over here at those plants that I thought could either be albidum or the subspecies parviflorum, and we see that, yeah, they look a lot like the subspecies parviflorum. So we can see that the floral scent gives us extra insight into the species identity. Now, rather than look at this all at once, I'm going to do this in parts. So we're going to do the left half first, because the left half represents the white sessile trillium. And we'll start up here with Trillium parviflorum. So these populations of Trillium parviflorum and those intermediates or questionable plants are all represented largely by these compounds linalool and a derivative called cis-linalool oxide. So you can see by the colors that those are the major compounds there. I'm going to drop down here to Trillium albidum without purple pigment, and hopefully you can see that this group of plants all has very similar floral scent compounds. We have citronellol, geraniol, benzyl alcohol, and caryophyllene. And the one reason why I want to go here is because these populations here represent the largest geographic range. And so we've got populations up here in the Bald Hills. We have populations in Oregon, and we've got populations in the Klamath Mountains at higher elevations, and even down in the Sierra Nevada foothills. So even with that large geographic range, the Trillium albidum without purple pigment group seems to have a consistency in their floral scent. And so I think that says something. Whereas if we go here to Trillium albidum with purple pigment, notice there's actually variation in there. So the plants with purple pigment, there's not really a consensus as to what their floral scents are. In fact, they seem to be a blend of the plants of parviflorum and albidum. We've got compounds from the parviflorum like linalool and linalool oxide, and we've got plant compounds from the Trillium albidum without purple pigment like citronellol and benzyl alcohol. So there's sort of a mix going on there. And then if we go down to Trillium albidum in the Marble Mountains, as I mentioned, they have a little bit of unique characteristics morphologically. And as you can see here, just by the colors of these pie charts, they've got different floral scents as well. They do have citronellol and geraniol like the other Trillium albidum without purple pigment, but notice their major compounds are citronellol acetate and geranyl acetate, these two compounds, which happen to be just esters of citronellol and geraniol. So they're not that much different, but they are in fact different. And they do present a different set of compounds in their floral scent. So there is some uniqueness to those populations. If we look now to the red sessile trillium, I'm going to start here with the simplest. So Trillium angustipetalum has the simplest floral scent of all of them. You can see it's mostly this compound isobutyl acetate with just a little bit of these other compounds. Those Napa County red sessiles also have a very similar floral scent, almost identical, except they do tend to have a few more trace compounds in them. Remember, one of our big question was, is Trillium kirbyashii a distinct species from angustipetalum, or are they the same species? Well, if we look at the floral scent compositions, look at Trillium kirbyashii. It's quite a bit different. In most of the populations, the major compound is this compound sulcotone, this maroon compound, whereas angustipetalum has more of the isobutyl acetate. So kirbyashii also has isobutyl acetate as one of its major compounds, but not quite the major compound in the majority of the populations. And not only that, it's got a ton of other compounds. So the other, can't find the cursor here. The other parts of the pieces of these pies are lots of other compounds in kirbyashii. So Trillium kirbyashii has the greatest complexity in its floral scent. It has the most compounds represented in its floral scent. So we can see that it does look quite a bit different from angustipetalum. And then if we go up here to chloropedilum, we see quite a bit of variation. So if we look at just variety chloropedilum, they're somewhat consistent with one another. The main compounds that they have are nerol, neral, geraniol, and draniol. And some of these compounds are very similar to what we saw in Trillium albidum. And this, I call this a quartet of oxygenated monoterpenes. There are 4 of these compounds. They're oxygenated monoterpenes, and they're typical of Trillium chloropedilum. So we typically see those compounds in the plants at these populations. However, if we look down here to chloropedilum variety giganteum, we see that a couple of the populations are similar to variety chloropedilum, but the other populations, they seem to have a lot of similarity with angustipetalum. So there's more variation there. They have more of that isobutyl acetate like the other red sessiles do, and they have less of those compounds that are typical of variety chloropedilum. So there's lots of variation within chloropedilum, and that makes some sense. Remember that petal color variation that we saw and petal shape variation with chloropedilum was pretty substantial, very dramatic. And we see that same variation reflected in the floral scent composition as well. So there seems to be a parallel between morphology and floral scent in terms of variation. So when there's high variability in the morphology, we also see high variability in the floral scent composition. So we just looked at the data in terms of populations and pie charts, but I'm going to present the data to you now in a slightly different way. And we're going to use some statistical analysis. And I didn't do these statistical analyses, rather Matt Riley did. And Matt Riley is a forest ecologist who works with the U.S. Forest Service up in Oregon. And he did these analyses. So in these analyses, how they're different is that each individual is represented here. So there is a symbol here for each individual. And what you'll notice is that their symbol is color-coded based on their species. So we've got Trillium albidum as the blue triangles and so forth. So I've got them separated according to species, or in the case of the Napa County red sessiles or those plants that I didn't identify as albidum parviflorum, I also color-coded those as well so we could distinguish those. So we're going to go through this just sort of piece by piece. And I first want to point out the low-hanging fruit here. So Trillium kirbyashii, it looks pretty distinct from the rest, right? It's clustered together. We can separate it from all the rest of the samples here. And when you look at these plots, what you want to think about is the fact that these analyses were done such that they're based on the 92 different compounds in the floral sense, and sort of their their relatedness to one another and the amount or the percentage of each of those compounds in the floral sense of each of these individuals. So these plots that I'm going to show you show relatedness of the individuals to one another based on those 92 floral scent compounds. So we can see here once again that Trillium Kurobayashi, it seems to be quite distinct from the rest of the Trillium. And remember, once again, one of those questions was, how does Trillium Kurobayashi compare to Angustipetalum? Are they different species or are they the same species? And based on floral scent, I think we could argue that these are distinct species. They occupy very different spaces in this plot, and they're also clustered together uniquely. If we look at those Napa County red sessiles that I said were very similar to Angustipetalum, but could also be Chloropedilum, look at those. So the black dots are Angustipetalum. They mesh in nicely with the orange dots, which are Angustipetalum. And so we've got the Napa County red sessiles, the Angustipetalum, all clustered here in this orange circle. But what you should also notice is that these pink triangles here, those represent Trillium Chloropedilum variety giganteum. Those also seem to cluster in this area as well. So there's still a question as to what those Napa County red sessiles are. If we look over here in the purple diamonds, we're going to see Trillium albidum subspecies parviflorum. So we see that the subspecies parviflorum clusters together nicely. And if we include those intermediates, or the plants that I couldn't identify as either albidum or parviflorum, they also cluster nicely with the parviflorum, except for a few of these that might extend out a little bit of a distance away. So we might also say that Trillium parviflorum could be a distinct species. Now, Trillium albidum, the rest of Trillium albidum, excluding parviflorum, is in this large circle here. And what I want to do is I want to look at this species a little bit more closely. And so we're going to look at a different plot here where the colors are a bit different. So we've got Trillium albidum separated into the 4 different groups. The plants from Marble Mountains are these red triangles. You can see they cluster together nicely. So populations of Trillium albidum in the Marble Mountains are all somewhat similar to one another. Down here, we see the populations or the individuals from Trillium parviflorum populations, in addition to those intermediates or those questionable plants. They all cluster together nicely. And then the green dots are Trillium albidum without purple pigment. There's sort of a large area that they occupy. And then those plants with purple pigment, as you can see, really are just scattered throughout everything. So the ones with purple pigment seem to have features that encompass those of all of the other varieties. So there's a lot of variation in plants with purple pigment, and then also even more in the plants without purple pigment. But parviflorum clusters nicely, and those plants in the Marble Mountains tend to cluster nicely as well. Now, about Trillium chloropedilum, we've got 2 varieties. Again, there's variety chloropedilum, and here I have in the green circle are those plants. So chloropedilum are the green triangles, and you can see they overlap extensively with Trillium albidum up here. And they also are in this intermediate region between Angustipetalum, which are the orange dots, and albidum, which are the blue diamonds. So chloropedilum kind of extends throughout albidum and then comes close to Angustipetalum. If we look at variety giganteum, it overlaps extensively with albidum. It overlaps extensively with Angustipetalum, and it also occupies this intermediate region. So I want to remind you that albidum and Angustipetalum, their regions are quite separate. They're distinct. But once we look at chloropedilum now overlaps with both albidum and Angustipetalum, and to a greater extent if we're looking at the variety giganteum. So I mentioned before that there's one theory that Trillium chloropedilum could be a hybrid or a hybrid swarm. And so if we were to consider it being a hybrid, perhaps the parents are Trillium albidum and Trillium angustipetalum, the maroon petaled plant down here and the white petaled plant up here. So that's one potential theory. And the data indicates that might be the case, whether we look at the statistical analysis here, or even when we looked at the pie charts, we also saw colors in those pie charts that were intermediate between those two species. So just to summarize here, the chemical compositions of these floral scents are largely distinct for each taxon. Even though there's some overlap, we can see that they enhance the set of characters that we can use for each of the species to help distinguish them. And we saw very distinct blends of floral scent composition for Trillium albidum subspecies parviflorum, or if we want to call it Trillium parviflorum. So maybe we could argue that that's a unique species. We also saw very different floral scent composition for Trillium kerbiaceae. Maybe we could argue that that's a unique species. Between Trillium kerbiaceae and Trillium angustipetalum, well, kerbiaceae had a very complex floral scent composition, whereas angustipetalum was very simple, and they were very distinct from one another. So we could argue that those two are in fact separate species. Both Trillium albidum and Trillium chloropetalum had large variation in their floral scents, and they also, as I pointed out, have large variation in their morphology. So we can see that the variation in floral scent couples to the variation in morphology, and so maybe there's a genetic connection to these traits. In terms of Trillium albidum, that high variability in both floral scent and morphology could be due to the fact that they occur over a large geographic range. It could be that they actually co-occur with all the other species. They co-occur with kerbiaceae, with angustipetalum, and with chloropetalum, depending on where they are. So there could be more than one explanation for why there's variability in Trillium albidum. And then as far as chloropetalum, that high variability in petal color and petal shape, in addition to the high variability in floral scent composition, could be due to the fact that it's a giant hybrid swarm. And that's one thing that maybe we could hint at with the floral scent composition, but I think to confirm that, we would need some genetic studies done. And that's not something that I would do, so a plant geneticist would have to confirm that. And I have collaborated with a plant geneticist at University of Tennessee in Knoxville, and with his methods, which he uses for the eastern sessile trillium and eastern pedicellate trillium, they don't work for the western sessiles. So even though it's been tried, we still don't have answers. And one reason for that could be that these plants have evolved more recently in evolutionary time. So there might not be the markers there that they're used to seeing to be able to distinguish them. So this work was recently published at the end of last year in the American Journal of Botany, along with two co-authors, Matt Riley, who, as I mentioned, did the statistical analysis, and Elena Petlevsky, who was one of my undergraduate researchers who worked with me for a couple of seasons and also did a large amount of analysis. And then Mihai Temescu, who's a botanist in our botany department here, and also his postdoc, Candela Blanco Moreno, they took my floral scent data and they carried out a phylogenetic analysis. So they treated the floral scent data much like a phylogeneticist would treat genetic data, DNA data, and they tried to explore speciation of this group of plants using that method. So this should be out in the next issue of Annals of Botany. So with that, I should mention actually that I started this project back in 2009. That was a long time ago. There's a lot of work with teaching, so there's not a whole lot of time to get out in the field and do research. But over that course of time, over many years, over a decade, lots of people were involved in helping. So I mentioned Matt Riley and Elena Petlevsky. Lots of conversations happened with people. Michael Messler, Rob Raguso. So Rob Raguso studies floral scents of Trillium in the eastern US, and he's a wealth of knowledge. Russ Graham, Larry Stouffer, their Trillium enthusiasts, and they grow Trillium, and so they've got lots of knowledge as well. And I mentioned Mihai and Candela. Robin Bensi in the herbarium, worked with her lots in getting specimens in the herbarium. Louis McCrigler, he kept my instrument running, the gas chromatograph mass spectrometer. He was instrumental in that. No pun intended there. And various other people, Lawrence Janeway, Brett Hall, James West, Dana York, and they were very helpful in finding populations. I should mention Ray Collette because he's now deceased, but he ran the UC Santa Cruz Arboretum for a number of years. And he was somebody who would always poke and prod professors at a variety of institutions across California saying, what's going on with these plants? Somebody please study them. Well, I finally asked somebody to introduce me to him, only to learn he'd passed 2 weeks prior to that in 2012. So I never got to meet him, but he's been an inspiration for this throughout. And I have used his plants for some additional studies that I didn't talk about here. And then another couple people whose private property, they allowed me to go on and study their plants. And Eli Callison was an undergraduate student who worked with me as well, who I see is here as well. And William Wood, or as known as Bill Wood, he introduced me to the methods that I use for this. And, I started this project before iNaturalist was a thing, or before I knew it was a thing. So it was very difficult to find populations. But with iNaturalist, it really helped to find more and more populations and gain more insight into the variation of Trillium and where they exist. Lots of parks and forests and lands where I sampled specimens, lots that aren't mentioned here where I didn't necessarily have permission, but you got to take that opportunity when it's there, because you never know if they're going to bloom the next year. And then, of course, all this research was carried out at Cal Poly Humboldt, formerly Humboldt State University. And before I finish, I just want to say, don't forget to smell the Trillium. So Trillium bloom anywhere between January and June, depending on the population, the elevation. So you've got, almost half the year of opportunity to smell these plants. And so I do highly recommend just bending over and smelling them when you see them in bloom. But note, their floral scents will be different as the flower ages. So all of my analysis took place in the early blooming period. So between the first day of blooming and maybe about day 6 of blooming. After that, the floral scent dissipates and it's not as strong. Some people might say some of these flowers smell fetid or smell bad. Well, it's because they're past their scent producing period. So they do have a finite period right after they bloom where they have those strong, vibrant floral scents. Okay. Questions?
You'll have to repeat the question because you didn't set up a microphone.
Okay.
When you're out in the field and you're doing more sending, say you're sending 20 of them, you get scent fatigue.
So the question is, do I get scent fatigue when I'm out in the field smelling the plants? Yes, I do. At the beginning of the season, which starts, as I mentioned, in January or February, depending on the population, I can't wait to smell them. And I'm so excited to smell the plants again. But after, especially Kerbiaceae, after smelling those over and over, after a while, I get sick of it. Trillium ovatum, the one in our local redwood forest, that's another one, over the season. I have a hard time smelling them over and over, but initially I love their scents, but they can get to be a little annoying over time. Yeah. Yeah.
And are any of these compounds, the typical compounds we talked about, easier or harder for plants to produce? Because it takes more energy to produce some compounds than others.
I'm sure, yeah. So the question was, are there some compounds that are easier or harder for the plants to produce? I don't know the exact answer to that question. I'm going to guess yes, but which ones yes, which ones no, I really couldn't say. And, a lot of these compounds are produced along different biochemical pathways. And, even though I showed you for Trillium albutum, and Trillium chloropetalum, there are these whole sets of of oxygenated monoterpenes. Well, they're all very similar in structure to one another. So it might be that, you know, this plant has this enzyme to say, oh, let's go along this pathway, whereas this plant has another enzyme to go along a slightly different pathway. So they all, you know, are very similar up until a certain point. And so they just have different enzymes that key for different compounds. But whether or not, which ones are harder or easier to produce, I couldn't answer that. Yeah. Yeah. Dana? Thanks for that question. Yeah, so the question is, what pollinators am I seeing? So one, you will probably be surprised to know that we still do not know who pollinates these plants. I have spent a lot of time trying to figure out the pollinators. Now, the types of insects that we see visiting these plants are not bees, they're actually beetles. So, and they're not large beetles, they're very tiny beetles, very, very tiny beetles. And there are a few different types of beetles. In fact, I've collected them, I've photographed them under a dissecting scope, and I'm gonna get somebody to identify them. Whether or not they're just there visiting the plants and munching at the pollen, or whether or not they're actually transferring pollen between plants, or whether or not these plants can self-pollinate, all these questions are still unanswered. People have tried to answer them. Michael Messler has done some studies with undergraduates in past years. The results are inconclusive. But once again, for these Western sessile Trillium, tiny beetles and a number of different species of these tiny beetles are the predominant visitors. And if there is a pollinator, they're the likely pollinators. Yeah. Question back there.
Did your specimens have to be like especially well preserved?
Yeah, so what I do is when I collect them in the field, I put them in a vial and close them up, and then I put them on ice or with ice packs. And so they pretty much have to stay with ice or ice packs or in a refrigerator until the time of analysis, and then I pull them out. Yeah. It depends. If I go up to Washington, then there might be a couple days. So to give you an idea of when I've gone up to Washington, so I drive up there on one day, I collected the next, and I drove home later that day. And then the next day I was in the lab analyzing. So I try to analyze them within two days, but sometimes, like when I've gone down to the Bay Area to collect chloropedilum, you know, it's a long way and there are lots of populations, so I sample from different ones. So sometimes it takes a number of days to get all those samples analyzed. So sometimes it could be up to 4 days, but if they're in the refrigerator, they seem to hold pretty well. And so it doesn't seem to be an issue. Yeah. Yeah.
This is far outside my field of studies.
No worries.
I was curious about the correlation between morphology and the chemical composition of the scents. So it's like the isobutyl acetate and the purple color.
Yeah.
So my question is, for the populations of the chloropedilum giganteum, there was 3 that are really high isobutyl acetate.
Yes.
I was wondering if you happen to notice a higher occurrence of the purple petal.
Yes, very good observation. So one thing I didn't mention was, you know, oh yes, sorry. So the question was, is there a correlation between the floral scent compounds and the petal color? Essentially, if I'm getting that right. Yeah. So one thing that one conclusion I didn't mention to you guys was that the red sessile Trillium tend to have more esters. So that isobutyl acetate, that orange compound, is an ester. And so the red sessile Trillium tend to have more esters. The white petaled Trillium tend to have more oxygenated monoterpenes. So things like citronellol and geraniol and nerol and geraniol. So yes, there is a difference. And in those populations of chloropedilum variety giganteum, those where I saw more of that isobutyl acetate, the compound that is more associated with the red petal plants, yes, those populations tend to have more of that, that burgundy color. So with chloropedilum giganteum, if you go to a population, you're going to find that the petal colors are from that burgundy to pink. So, and you know, everywhere in between. Or you'll find that the populations are from pink to white. And so there does seem to be a difference in those petal colors, and the pink to white tends not to have that isobutyl acetate, and the ones with burgundy or dark red to pink tend to have more of that isobutyl acetate compound, the one that's associated with the red petaled plants. Yes, good observation. Mhm.
I was just curious if anyone's done clustering like you did.
I'm not familiar with morphometrics, but exactly. I'm not a statistician at all. But this study right here, so Mihai and Candela took my data and they did a phylogenetic analysis. So they're, typical, what you would see in terms of trees and clusterings that you would see with genetic data. But Candela also did some different statistical analyses as well. And the exact names of those I can't remember offhand, but she compared her statistical analysis and the phylogenetic analysis with the non-metric multidimensional, can't remember the last word, analysis that we presented in the American Journal of Botany paper. So this data has been analyzed to a great extent. But whether or not your specific one has been used, I couldn't address that exactly. Yeah. Yeah. But maybe you mentioned morphometrics. So maybe you're talking about morphology compared to floral scent.
Yeah, based on the. Yeah.
Yeah, unfortunately, the best I've been able to do in that sense is I take lots of photos. And so, when I come back and see the floral scent composition, I can say, oh yeah, this population has these characteristics, but I never did any measuring. That would require a lot more time that I had, because again, to preserve that floral scent and to make sure everything was cared for, I had to get it in the cold very quickly. So I couldn't always spend a whole lot of time. So yeah, lots of pictures taken quickly. So I do have a general sense for each population. I've got extensive photos, but no measurements. Yeah. Other questions? Yeah.
Are there instances where taxonomy has been resolved in floral scent, or is this like?
So the question is, are there instances where taxonomy has been resolved using floral scent? And we do mention a couple of articles that do try to address that in this Annals of Botany paper. I can't remember offhand how many or to what extent, but I'd say more often I've seen where studies look at different species and show that they have different floral scents, or maybe different color morphs, or different color morphs, and they might have different floral scents. And one reason to do that is to look to see if there's a barrier to pollination. But there aren't many studies where they've tried to use floral scent to distinguish species. Yeah. Other questions?
Thank you.
All right. Thank you.